All right, so now let's talk about our antifungal agents. And so again, these are going to be agents that are we've had them for use for a very long time. There's not much research on new antifungal agents because they work very well, and fungi do not mutate similar to, or they do not acquire antifungal genes like we see in our bacteria, for example. And so the arsenal that we have for fungi are relatively good. And so we're going to talk about our polyenes, which are most famous as amphotericin B, our azoles, echinocandins, anti-metabolites, griseofulvin, for example, all of those. And so we know that our fungus is going to be a eukaryotic cell, but there are some targets that are going to be important. Our cell wall, again, we have a chitin and our glucan synthesis that you can target. Our cell membrane, which is our ergosterol synthesis, so those are their own version of cholesterol, and that's where our azoles will target. Now, for the other ones, you don't really want to target protein synthesis too much. That they have a few that are going to target protein synthesis. We have some metabolites that will target different metabolisms. We have microtubules. Griseofulvin is going to target microtubules. And we have our polyenes, which will lead to direct damage of the cell membrane. Now again, most of the resistance is going to be from our Candida species. Others are thought not to be very resistant. There's no evidence that the fungi have any enzymes that can neutralize the antifungal agents. They do, however, have efflux pumps similar to our bacteria. They can also have mutations in their targets, so they can alter their targets similar to the way that bacteria can alter the penicillin binding proteins. And then they can reduce access to the drug targets by compartmentalization, back to that efflux pumps, and ways such as that. So our polyenes are going to be important for treating life-threatening mycosis. So when we talk about opportunistic mycosis, many of the patients are going to be treated with amphotericin B. So amphotericin B has different formulations. Some are going to be less toxic, so you can keep them on as a pre-exposure prophylactic type therapy. Some of them, though, are going to have some toxicity. So nephrotoxicity, fever, chills, hypotension, all are side effects of amphotericin B. Amphotericin B is going to affect ergosterol, which is a type of cholesterol. So the toxicities that can bind to cholesterol and cell membranes. And some people don't understand, but cholesterol and cell membranes is critical for our cell membranes to actually function properly. Another polyene is nystatin, and this is a topical agent, so you do not ingest nystatin. So what are the polyenes really doing? The polyenes are basically binding ergosterol, and they're damaging the membrane. So you can think of them as sort of like a defensive, punching holes into the membrane of your fungal agents. Complement, punching holes. So it's kind of the same sort of thing. It's going to produce an ion channel. You're going to have a flood of electrolytes through that. You get osmotic integrity destroyed, and eventually the fungal cell will die. Resistance is going to be uncommon in our fungi. The azoles are going to be another class that's going to be highly used, and these are going to inhibit a fungal protein or enzyme, is a P450 dependent enzyme that's called lanosterol 14-alpha demethylase. And this is important for converting lanosterol to ergosterol. So while we have our polyenes that are punching holes in our fungal cell membrane by targeting ergosterol, we have our azoles, which are trying to prevent the production of our ergosterol. So this is the metabolic pathway for your fungi to actually generate ergosterol. You're going to start with our squalene, which if you've had biochemistry, you know that we use squalene as a precursor for cholesterol. So our fungals are a eukaryote, so they use the same precursor. So for squalene to become ergosterol, it has to go through several steps. And so we have several different types of antifungals that will block this. The azoles, which we're talking about right now, is going to block lanosterol from becoming ergosterol. And it targets the enzyme. It does not target the actual cholesterol. So because it targets the enzyme, this is not going to target the host enzyme. So this is an enzyme for the fungus, 14-alpha demethylase. And so it's going to prevent ergosterol formation. So this is going to be fungostatic in our Candida species and our neoformans, Cryptococcus neoformans. It's going to be fungicidal in Aspergillus species. Now there are a few different azoles. We have imidazole, which has 2 nitrogens in the azole ring, and we have triazoles, which have 3 nitrogens in the azole ring. And the key is these are going to have lower toxicity and lower protein binding, so overall you can use your triazoles longer than the imidazoles. Mechanisms for resistance, these are going to be rare again, but Candida does have some azole resistance, especially for the cruzi, for example. They are able to change their target enzyme. Lanosterol 14-alpha demethylase is going to be coded by a gene called ERG11. So these Candida species can gain resistance through mutations in ERG11. So the enzyme is less susceptible to being targeted. Or you can have overexpression. So that means that the fungi now makes too much of the enzyme, and so you have to increase the dose of the azole to overcome that. You also have some that can upregulate what we call ATP binding cassette transporters. These transporters are sort of medicine pumps, you can think of them. And so they pump out a lot of different medicines. And so they can pump out the azoles, for example. And so that's going to increase the egress. They also have another egress pump called the MDR, major type efflux pump. And that is going to, again, cause the egress or the pumping out of the drugs. Aspergillus can be resistant through drug efflux or altering that target enzyme. So the target enzyme, of course, here is the lanosterol 14-alpha demethylase. Cryptococcus neoformans overexpresses the MDR efflux pump, as I say here, and also has the CDR efflux pump. Echinocandins are going to be inhibit the 1,3-beta-glucans, which is a key fungal cell wall component. And so this is going to be toxic to fungi that are going to need this. So it's going to be fungicidal, killing Candida species, static against Aspergillus species. The echinocandins are going to have to be delivered through IV. You cannot ingest them. And there is some resistance, and it's always going to be associated with prophylactic long-term use of this particular antifungal. We have some anti-metabolites. We have flucytosine, and this is the antifungal agent, and it's going to be a pyrimidine analog. That's one of our base pairs for RNA DNA. And basically, you're going to have deamination in the cytoplasm of the fungi of your flora uracete. So it's a deamination. So basically, you're going to have this pyrimidine analog. It's going to compete with uracil, and therefore it's targeting RNA synthesis, because remember, uracil is only an RNA, not in DNA. The RNA inhibits protein synthesis, and this is going to have some activity against these species listed here. Now again, resistance to these antimetabolites is going to be very rare. Candida is going to show the most resistance, and neoformans, especially if you only use the antimetabolite as a monotherapy. Typically, an antimetabolite would be given with one of those other azoles, amphotericin B, etc. Now, one thing to keep in mind is it has no activity against some species, such as Rhizos mucormycetes. Aspergillus has no activity against, nor are non-pigmented molds, those hyaline molds. And then we have our allylamines. Allylamines go back to the ergosterol story, and so they are going to work upstream of the azoles and polyenes, and they're going to prevent the conversion of squalene to lanosterol. An example of an allylamine is a turbinafine. This is going to have broad spectrum activity. It's going to be systemic, so it can be important for treating dissemination. It is going to be important in treatment of dermatomycoses as well as those of the nails, onychomycosis, and has very few side effects. There is some resistance with a multi-drug efflux pump. That is that CDR1. Griseofulvin is going to be an oral agent. It's going to inhibit mitosis, and it interacts with the microtubules of the fungi, so it basically does not allow the microtubules to form, so you can't get division and no growth. It's oral, but can be used to treat dermatophytes as well. Itraconazole and terbinafine are more rapid, so griseofulvin is a second-line agent, and it does have some side effects associated with it because it is binding to microtubules. Of course, we have microtubules as well.